At this point, we're going to shift to an even more detailed level to describe inhibition and excitation of neurons, generation of action potentials, and synaptic weight change processes in terms of chemistry. Action potentials, as I mentioned before, are spikes of electrical voltage supported by chemical processes. These chemical processes involve ions, and ions are electrically charged atoms or small groups of atoms. To start, we need to go back to the cell membrane. As I mentioned earlier, the cell membrane is impermeable to most types of atoms and molecules, and specifically, it's impermeable to ions. But again, as mentioned earlier, embedded in the membrane are large numbers of protein molecules. And one important type of protein molecule is the sodium-potassium pump molecule illustrated. This molecule pumps sodium and potassium ions in opposite directions across the membrane. It has two units, shown in this more conceptual drawing. The alpha subunit binds to sodium and potassium ions, but the beta unit is critical for the operation of the pump. But for simplicity, I'll leave out the beta structure. The sodium-potassium pump shifts between two configurations. In the configuration in this slide, 3 sodium ions in the interior of the neuron bind to 3 specific sites on the pump. At that point, the pump has an affinity for adenosine triphosphate, or ATP, which is the universal source of chemical energy in biology. The ATP breaks to form ADP, adding energy to the pump by leaving a phosphate group attached. The energy release changes the conformation of the pump to face the exterior of the neuron, and also breaking the bonding of the sodium ions. The sodium ions are therefore released outside the neuron. In this configuration, the pump has an affinity for potassium ions, which therefore bind to the 2 receptor sites. The binding of the potassium ions results in a reverse change to the pump conformation and release of the phosphate. These changes also break the bonding of the potassium ions that are therefore released into the interior of the neuron. So at this point, the pump protein has returned to its original state, and the cycle can repeat. The effect of the pump is therefore to create a deficit of sodium ions and an excess of potassium ions in the interior of the neuron relative to the outside. Because 3 sodium ions with single positive charges are moved outside, for every 2 potassium ions, again with single positive charges, moving inside, there's also an electrical voltage difference that's created across the cell membrane, with the inside more negative than the outside. There are other ion pump proteins that maintain the inside of the neuron with a relatively low level of negative chloride ions, a relatively high level of negative bicarbonate ions, and an extremely low level of positive calcium ions. The overall effect is that the inside of the membrane is about 70 millivolts negative to the outside, and most of this difference is actually maintained by the sodium and potassium ions. This potential difference is known as the resting potential, because it's the potential to which the membrane tends to return if it's disturbed in some way. Now, although the membrane is impermeable to all these ions, it's also penetrated by various proteins that in some situations can serve as channels for the ions to flow through. An ion channel usually only allows passage of one type of ion, generally sodium, potassium, calcium, or chloride, but sometimes it allows more than one, provided they have the same charge. Most of the time these channels are closed. And if they're open, they don't pump ions, all they do is allow ions to flow, and the ions will tend to flow from a region of high concentration to a region of low concentration. In specific circumstances, the ion channel can open and allow passage of the ion type. And when the channel's open, it allows ions to diffuse through at a certain rate. And as I said, it doesn't pump the ions, but if there is a concentration difference between the inside and the outside of the membrane, there'll be a flow of ions from the region of high concentration to the region of low concentration. So in the case of sodium ions, they flow from the outside to the inside. And because the ions carry an electrical charge, this flow is an electrical current that disturbs the resting potential. So opening a sodium channel makes the inside of the membrane more positive in the vicinity of the channel. So the magnitude of the potential difference in that location is reduced. That kind of reduction is called a depolarization. The flow of ions is fairly small, and only the concentration close to the membrane is affected. If the channel closes again, the excess sodium ions diffuse away from the membrane towards the interior of the neuron. The membrane potential then will, of course, return to its resting value. Opening a potassium channel will make the interior more negative because positive potassium ions flow outwards, increasing the magnitude of the membrane potential, in other words, making it more negative. These increases are called hyperpolarizations. Opening a chloride channel will also hyperpolarize the membrane because the negative chloride ions flow inwards. But opening a bicarbonate channel depolarizes the membrane because the negative bicarbonate ions flow outwards, which has the same effect as an inward flow of positive ions. Now, when will an ion channel open? Well, there's two types of mechanism. In one mechanism, the channel opens when a chemical available outside the neuron binds to it. The opening lasts for some period of time, and then the chemical unbinds. And these chemicals are called neurotransmitters, and they're generally released at synapses by the presynaptic neuron in response to an action potential. Chemical processes in the environment are constantly removing the neurotransmitter, so the channels only stay open for a short period. The other mechanism is that a channel opens when the membrane potential is pushed away from the resting potential and gets to a value within some specific range. For example, a channel may open if the membrane is a bit depolarized. In other words, if the inside of the membrane is a bit more positive than the resting value. As a general rule, most ion channels on axons are voltage gated, but a significant proportion on dendrites are neurochemically gated. There are lots of different types of ion channel, each one opening by a different mechanism, passing different ions, and with different channel dynamics, like the length of time they remain open. Any one ion channel molecule alone carries a very small current, but different areas of the neuron membrane have high concentrations of different types of ion channels, and these concentrations determine the function performed by that membrane area. These are some very common ion channels in the brain. The AMPA channel and the GABA-A channel are opened by neurotransmitters. The NAV, KCNQ and CAV2 channels are opened by changes in membrane potential. And the NMDA channel it will only open if there's a combination of both. As we'll discuss, the AMPA channel is the primary channel responsible for the excitatory effects of the glutamate neurotransmitter. The NMDA channel also responding to glutamate, is critical to changes in synaptic weights. The GABA channel is the primary inhibitory channel, and the NAV and the KCHQ channels are critical to the propagation of action potentials along axons. And the CAV2 channel is critical for translating between an incoming action potential and the release of neurotransmitter. But there are lots of other ion channels, each with its own important function.